So this is going to be a lesson looking at meiosis, which is, as it says, reductive division. So we have our original cell that is going to be a germline cell. Notice that we have pairs of homologous chromosomes. Each set of chromosomes is a different color. Before meiosis happens, the cell is going to actually go through a round of DNA replication, and it will end up going through basically an S phase, after which you will have these nice duplicated chromosomes. So here you have a pair of homologous chromosomes, but keep in mind you also have each of these chromosomes exists as a pair of sister chromatids. The chromatids are identical, the homologous chromosomes are not identical. Also, in important structures, we do have the centrioles there, which we'll talk about in more detail in a moment. Now, before prophase starts, this DNA is actually not identifiable as those stereotypical X-shaped chromosomes. Instead, it is a kind of loopy mass of stuff called chromatin, and it is not going to be distinguishable. During prophase, what happens is the nuclear membrane is going to break down. You have centrioles that are going to replicate so that you'll have two sets of them. And you now have your basically pairs of sister chromatids that are going to be grouped up in pairs of homologous chromosomes. The centrioles at this point are going to start making the protein spindle, and you're going to have those chromosomes condensed so that you'll see them as distinct shapes. What I don't show you here is that those homologous chromosomes will pair up with each other. Once they've paired up with each other, they're going to stay paired throughout the remainder of meiosis 1. So you'll notice we have things lining up kind of along the center of the cell, and so that should hopefully give you a clue that we are going into metaphase. In this case, it's metaphase 1, where we're going to have pairs of sister chromatids lined up at the center of the cell. Notice that you do have overlap between the homologous chromosomes. That's where crossing over is going to occur. And also note that we have a metaphase plate there. A metaphase plate is not an actual physical structure inside of a cell. Instead, it's just kind of the center of the cell where the pairs of homologous chromosomes are lining up. So you've got homologous chromosomes having crossing over, they're recombining with each other, and then you're going to move on to the next stage, which is anaphase 1. Notice that the cell is getting longer. It is going to be basically growing here, so that way it will be able to split into two separate cells. So we're already preparing for cytokinesis. And apologies for the redrawing. It's sometimes difficult to remember drawing recombinant chromosomes. All right. So you'll notice that I have these chromosomes that are basically part blue, part pink. Some of them come from one set of chromosomes, and then part comes from another set of chromosomes. So this is anaphase 1. We are separating homologous chromosomes from each other, and they're being pulled to opposite ends of the cell. Now we're moving on to telophase with my lumpy cells. Our centrioles are at opposite ends still. We don't have the protein spindle anymore. It's kind of broken down because we've pulled our chromosomes, our homologous chromosomes, to opposite ends of the cell. And kind of see them separated there. And this is telophase 1. At the same time as telophase 1 is happening, you're going to have cytokinesis happen as well. But at this point, since you've now separated your homologous chromosomes from each other, you are now creating a haploid cell from what was a diploid cell. So from this point on, you will be having the two cells that have been created at the end of meiosis 1, Each of them is going to be going through the process of meiosis 2, and generally cells will go straight into prophase 2. So chromosomes will not decondense or anything like that. Instead, they will just start lining up for a second division. So we've got another set of two centriole sets that are there. We've got now our set of chromosomes in each new cell. And sorry, it takes some time to draw it. And you're making a new protein spindle because you're going to be separating things again. And then we're ready to move on to the next stage, which is metaphase 2. So remember, in metaphase, you are lining things up along the center of the cell, along a hypothetical metaphase plate. By the way, sorry that things keep popping up. This was my first time trying to do a screen capture on my tablet. So notice things are lining up at the center. Notice we still also have those kind of double-colored chromosomes where we've exchanged bits between homologous chromosomes, and that's going to be maintained throughout. And now we have these chromosomes, they're lined up at the center. It's pairs of sister chromatids that are lined up, and they are going to be attached at their centromeres to these fibers of the protein spindle. Then we move on to the next phase, which is anaphase 2. Our cells are elongating again because they're getting ready for a division. We still have our centrioles and our protein spindle fibers. But now we're pulling apart those sister chromatids from each other. Notice we still have those recombinant chromosomes there. And these guys are getting pulled apart or dragged apart by the fibers of the protein spindle. So the spindle fibers shorten. When they shorten, that pulls sister chromatids apart from each other, and the cells elongate until we get into now telophase 2. And during telophase 2, what's going to happen is you're going to have cytokinesis happening at the same time, and you're going to separate into each of those cells that was there. It's going to be separated into two new cells, each of which is going to contain one set of chromosomes, unduplicated. So we separated first our homologous chromosomes, and now we're separating our sister chromatids. So we've created 4 new cells, and they're going to reform nuclei at this point. Protein spindles are going to break down, and you're going to have cytokinesis happen at the same time. So your end result is going to be these 4 new gametes that are created, each of which is going to contain a different mixture of genes in the different chromosome sets that we have there. So each of these gametes that's being produced is genetically unique, and they will have some mix of recombinant and non-recombinant chromosomes.